We're going to move now to the next speaker, who is Winston Yan, president of N=1 Collaborative. And Winston will talk to us about individualized medicine, or sometimes we call that precision medicine, needed for collective learning, and building an infrastructure for knowledge sharing at the N=1 Collaborative. So we have your slide on the screen now. Hello, Winston.
Hey there, can you hear me all right?
Yes.
Fantastic. Great. So it's a great honor to be here on Rare Disease Day. And then I think the second honor is just to be able to speak with all of the esteemed folks, both in the first session, first part of the day, as well as now getting to speak after Tim and so many other folks working in this space. Just making sure AV check, you guys can hear me and you can see the slides okay, yeah?
Yes, perfect, Winston. Yes.
I'll take that as great. So Tim really teed up the work that now we do at the N=1 Collaborative really well. And today I'm going to talk about how do we continue that process that he talked about of learning from these cases and proceeding forward with the proper rigor, but also urgency to bring these ultra rare disease treatments to all. And so my name is Winston and I help run the N=1 Collaborative, but in the truest sense of the word, it is a collaborative where we have so many of the investigators who have joined who have actually built these N=1 treatments in our mix. We have experts in industry, we have patient advocates, patients themselves, and drug developers, and we're all working to try and build a pathway to making this more routine. So let me tell you today about, I'll set a bit more broader context, and it comes from my background as I'm a physician scientist, but my PhD was in gene editing therapeutics, and particularly in CRISPR. And I want to set the stage that this is really a unique time for us in what we call platform drug development. The way I mean platforms is that, this is the dawn of an era where drugs can be truly programmable. And what that means for the ultra rare disease space is that you're no longer, you're able to piggyback on the work that's been done to develop, billion-dollar drugs for larger populations, and then use all of that knowledge to reprogram it and develop N=few or N=1 treatments for these ultra rare diseases. So here you have Spinraza. That was just a work that's I say truly epic in its impact and the precedents that it set from Ionis. That was an antisense oligonucleotide for the treatment of spinal muscular atrophy, and that was approved in 2016. That was an antisense oligo that could then be reprogrammed to become Milasen, and enable all of the work that we do today in 2018. And fundamentally, why that's important is that we're taking many of the pieces that are the same and just targeting it to a different genetically identified mutation. So this is the slide that I took from Tim in one of his early symposia, where he was describing just the similarities of nusinersen, also of Spinraza, and the delivery from Milasen. What this enabled was a much quicker development route. If you look at the sequences, right, nusinersen is on top, and then Milasen is on the bottom. We're getting into the territory of thinking this is deceptively simple, right? I just go on a typewriter and type out these aspects, and suddenly you have a new drug. It's not quite like that, and I'll go through the nuances of why we need to have collective learnings and be very active in how we're evaluating these and learning from these individualized treatments. But in the case of drug development, at least having the technology that you can program like this, it's already just a big step up from where we are with small molecules or even antibodies. So what happens today is that this is a graphical explanation of what I talked about with that piggybacking on the commercially fueled drug development. So on the bottom, you have the base of the pyramid that's built through hundreds of millions of dollars spent on clinical development by biotechs, by biopharma companies, VC money being poured in, and you learn so much about the platform for these larger indications. You learn things like the distribution, the toxicity, the immunogenicity. You learn how to make it in a really safe and clean way to go into patients. So what we can do with NFU medications is build on top of that. You can reprogram that and then reevaluate it with that new target. Of course, there will be sequence-specific assays that you'll have to develop, and you'll have to figure out what meaningful clinical markers or your on-target engagement will be. But fundamentally, you're learning from that broad base that's been established through the work of industry. So this is where, again, I'm emphasizing a lot of this collaboration. It's not only amongst patients and academics, but also just we stand on the shoulder of giants and that expertise that's, that deep expertise within industry. So being a gene editor, right, I look at Spinraza and I see Millicent, and Tim mentioned all of the other new antisense oligo programs that have come out. Well, there's also CRISPR that's on the horizon. And there are a number of trials that are in different organ systems with gene editing that really could enable the base of the pyramid to develop new gene editing treatments in various organ systems. So Tim mentioned this, and I thought that it is something to make sure that we are incredibly thoughtful about acknowledging that with progress comes challenges. And this was the New York Times article that he was referring to about how in the two patients, both of them for the KCNT1 epilepsy treated with the ASO, both of them had developed hydrocephalus. And this is an incredible, I feel like it's incredible to call out the poignancy and the challenge of being an investigator like Tim at this time, of running these trials, venturing into the unknown to treat patients, and then having this, I would say, an incredibly lonely space of saying, I don't have a randomized controlled trial of 10,000 patients worth of data that I can treat. I'm pushing the boundaries of medicine, and how do I infer and get information as much as I can to build the safest path forward for my patients? And so luckily he had the collaboration of folks at Roche. He had other investigators in the field to draw upon. But as a general field, the New York Times article outlined this need, that right now, so many of the scientists who are running these trials, you're not required to share information with one another, right? You can publish because it might advance your career or it's interesting to package all up, but fundamentally, you're not being able to communicate in real time and alter your clinical trials or your measurements in response to what others are seeing. And so I'll read this here because I think it is very fundamental to where we are today, that this is a new field of medicine. How do we share data and monitor so that we can learn from events and make this safer and potentially benefit future patients? This is what I will focus on today and how we're helping to support that at the N of 1 Collaborative. So if we take a step back, right, this is summarizing the need here. Today is Rare Disease Day. We've established that it's a misnomer, right? Rare diseases might be individually rare, but collectively they're so common. But the fact that you have that heterogeneity means that expertise often falls within silos, and there's not this infrastructure to communicate between them. And furthermore, for clinicians who are actively treating these patients, and particularly in using experimental treatments, you're not able to react quickly to these new developments because it's just of that heterogeneous landscape. Meanwhile, the very fact that you have these platform drugs is that you should be able to learn about their experiences as a class as you develop more individualized treatments, right? So there is the potential of learning from all of this in aggregate. So that puts up this need for the centralized system. There's a limited number of N of 1 trials right now. We really need to have that real-time sharing so that when investigators like Tim, like John, people who move these trials forward, when they're in the trenches and trying to figure out what's best for their patients, they can access that broader database to make informed decisions. And then why we do this is that ultimately we want to make sure the drugs are safe. We're also making sure that we're learning and making the drugs more efficacious. This means not only looking at the clinical outcomes, but we can pair that with the preclinical designs to know, what worked, what didn't work so well. How did activity in a Petri dish actually translate to the clinical activities? Can we just actually learn from each cycle of individualized drug development versus having redundant work that just wastes everyone's time? And ultimately, that's what's needed to fulfill our dream of going from millions to millions. So many patients are waiting, right? And if we don't make this process routine, standardized, and rigorous, it will continue to be a very bespoke avenue that people pursue, and it will not be actually the standard of care for ultra-rare diseases. One thing I want to emphasize is that real-time sharing does not equal publication. And I think there's this instinct that people say, well, I'll publish, right? Why not? Isn't that how academics share? And I want to emphasize here, it's this for large N clinical trials, for maybe competitive drug development efforts. Yes, controlling the publication, it almost becomes a part of your marketing campaign of saying, hey, look at our results. It's carefully digested, and it's in some ways it's it's displayed for the world. We're talking right now about sharing the real-time data so you can make clinical decisions in the best informed way. And that doesn't mean having it all published. And I think that is far too slow to make the decisions we need in the time to affect patients. So let's talk about building that learning platform at the N-of-1 Collaborative. Some of you are familiar with it, but others, this might be the first time you've heard of it. So this is a group of investigators, patients, drug developers, clinicians, scientists. All of us are here to develop that pathway in a repeatable, rigorous, and responsible way. What we want to do is to first aggregate the best practices, the data, and then we want to share. We want to make sure that this is a place where people can come back to as both a forum that we've convened for active discussion, as well as just lasting resources that we can share with the rest of the world. The values that we hold, we have to be rigorous, we're independent, and not beholden to any biases, and that we are inclusive to make sure that this pathway that we develop actually ends up being the rigorous and accessible to all. So where we think about our work is across a couple of different work groups. And this is where it's not, for us, it's not sufficient just to convene people, but we actually want to build those lasting products that I mentioned. And our work spans everywhere from how do you identify patients and see whether they're amenable to these ASO or maybe in the future gene editing therapies. Does their disease pathophysiology make sense for that? How do you design preclinical or do preclinical design, which is actually designing the molecule and running the right assays for it? How can we measure the clinical outcomes that are actually meaningful to both doctors as well as you as parents and the family? And then I talked about data sharing, right? How do we go beyond the N-of-1 and actually collaboratively share the collective learnings? And then finally, another thing that's often overlooked is how do we actually make this happen at a given institution? Because a lot of this N-of-1 trial infrastructure and know-how is new to even the most advanced, many of the advanced academic medical centers throughout the country. So are there template forms or protocols, consent forms, IRB structures that we can provide as here are some best practices that you can riff off of for making this happen at your institution? So today I want to focus on the data coordination work group, which as I described is about how do we collectively gather and then share that information. So we thought a lot about this, and it really takes a multi-part approach. The first is that we have to build the movement. I'm speaking to you today, sharing why we're doing this, but it's something that needs a broader recognition of how data sharing is different than publication, and how at this time and place, there's a renewed need for it in a more structured way than ever before. The second is we need to translate that momentum into actual building of the infrastructure. And this means figuring out how to structure a database, the protocols, to enable both that preclinical and clinical data sharing. And then the learning and the actual evaluation will come once we've populated a database with more and more N-of-1 trials. We get to see what works, what doesn't, as well as change the structure of things to better fit our purpose. So the first thing, as I mentioned, is about building that movement. And I know some of you have already signed this of the N1C recently, we released on change.org, open letter for data sharing. And this really articulates why we think it is so critically important in ultra-rare diseases to share data openly. If you have a phone, or I can send the link afterwards. This is a QR code for you to read the letter, see if you agree with it, and really sign or assign your name to this broader movement. I know so many of you that you might be saying, all right, how do signatures actually translate into real action? For us, the signatures actually give us leads into institutions and academic medical centers, as well as companies, to help us say, hey, you have members of your organization who are committed to data sharing. Can we build that pre-competitive data sharing agreement between you and us to help enable that? So your signature does matter in getting from signature and the petition to actual progress. The second thing that we've talked about is what types of data do we want to share? First, if we go from bottom to up, or from the blue square to the red, we want to think about what are protocols and best practices to help us get the best data. So many of you know that, this idea of garbage in, garbage out. And if we don't collect data in the right way or with the proper rigor, it ends up being not too useful for us to analyze downstream. So establishing the tools and best practices is a critical piece of making sure that our data is valuable at the end. And then on top of that, there's preclinical data, and then there's clinical data. So here we expand a little bit more on what we mean by each of those. So at the bottom, we have these protocols that will be broadly shared for both drug development, or the preclinical assays, as well as the actual clinical evaluation. This enables that harmonization and aggregation. And then also it allows us to build tools, whether it's of data analysis or just streamlining your workflow, that we can share back with the community. On top of that, we have the drug designs themselves. And this is where it's important for us to highlight we're sharing both effective designs and ineffective designs. Wouldn't it be incredibly frustrating to use the hard-earned money that you have raised or collected on experiments that people have already tried and shown that there's a toxicity and off-target effect from a particular ASO? Wouldn't it be great if there were a database that could show us where they are, and then furthermore, where we can learn from that in the future to pick out the features of particular ASOs or whatever modality it might be that makes it more likely to work. And then finally, on top of that, we have clinical outcomes. And this is really I would say, part of the crown jewel of what we want to offer, is because these clinical outcomes really help us determine whether a treatment or not is effective. It also helps us understand the safety profiles and make that informed balance of saying, is this, what is the real benefit risk? And is this something that is worth continuing? So I'll go through a couple of use cases, and this was put together by our team at the N-of-1 Collaborative. Imagine yourself in the shoes of an investigator who's running an N-of-1 trial. First, what if your patient they tell you that they're feeling euphoric after receiving their third dose of an N-of-1 ASO. You're thinking to yourself, was this a side effect of the anesthesia? Should I continue? Is it something that's causing some, broader, mental status changes? Does the dosing plan need to change? Do you need to order any additional tests to follow this up? These are all things that are difficult to really answer in the N-of-1 setting, and having the multiple layers of either protocols or lived experiences or even just the community to share things with are important. Then say after the 5th dose, your patient develops leg weakness. Thankfully, it's temporary, but you think it was drug related. Well, how do you continue then, right? Is there a lower dose you want to use? Are there class effects that you need to be aware of that you can consult the N-of-1 database, right? And then could you have predicted any of this before? Should you already have gone in eyes wide open to different clinical measures of safety that you needed to incorporate into your trial? And then importantly, are there other people running trials in the same organ system, using the same modalities that should be aware of this so they can change up the protocols for their patient? And then clinical outcome measures. I think many of times the clinical outcomes that are used for more common neurologic diseases, the ultra rares just aren't even on that same spectrum. And then furthermore, when you think about how do you measure, without the benefit of randomized controlled trials and large N's, how do you measure change to say that your drug actually did what you think it did? So those are important use cases as well as data to share with the community. And lastly, we think about this of saying, well, many of you are bundled into a disease by the gene that's knocked out, but actually every single allele within that may have a different profile. And so how do you treat those different disease-causing mutations? And how may they respond differently? This is an important part of building the case for whether it's a basket or umbrella, whatever you want to call it. It just means that we need the information first of how these individual mutations will respond to our treatment. So we've gotten to work and we started with preclinical data, and that was intentional. What we've done is go out and partnered with a number, or actually not partnered, we signed these data sharing agreements between the N-of-1 Collaborative and the, anywhere from Boston Children's to the National University of Singapore to UMass to the NIH. And we've signed these preclinical data sharing agreements knowing full well that it's a foot in the door to ultimately getting the clinical data sharing agreements, which have a lot more complexity around it. And here's where we ask you, if you're working with an investigator, could you gently nudge them or ask them, have you considered sharing your data preclinical and then later on clinical with the N-of-1 Collaborative? I should have put my email on here, but please do reach out to me. I'm just winston@n1collaborative.org, and we can make sure that there is a ready and willing party on our end to follow up on that interest on your end. And from our perspective, it's going from that pledge to progress means there's very specific builds that we need to do from the preclinical side, these blue bars up here, to the clinical database, to actually then how do we implement this and publish our work and share that knowledge with the rest of the community. I won't go through all the details here, but know that all of these take work. And again, we are actively in the build phase. We need volunteers and we need funding and support to make sure that this can happen in a sustainable way. So in the last couple of minutes of the talk, I'll just go back to the bigger picture. For me, the ultimate goal is that we're starting up with Mila, and we want to end up in a place where anyone who has that orphan, whether it's rare, ultra rare, N-of-1, however you want to name it, anyone who has that genetic disease, you can receive a bespoke treatment as that standard of care. To me, learning about drug development in my other life in biotech is that when you work backwards from that goal, how do you end up making that scale happen? Well, you need to think about things like reimbursement and who's paying for it. You need to think about equity and accessibility, because if you don't scale it, then only the people who can raise $2 million are going to be able to afford these life-saving treatments. Working backwards from that, convincing the regulators, or sorry, convincing the payers to reimburse it usually means that there is some sort of regulatory body that says this is a process that's good to go and we approve it. In our case, it may not be a product itself, but it's just a process of doing this entire N of 1 individualized treatment development. Well, to get to regulatory acceptance, that requires good data. And you need to rigorously establish that efficacy, that benefit risk, so that there's beyond a shadow of a doubt that these treatments are actually doing things for patients. And that comes from building a system from the beginning that is repeatable, that can be standardized, and that we're actively learning from all the time. It's still early. We are just here in our journey, but that's why I think there is a need for us to actually build and create the system to do it right. And to do so means that sharing and the collaborative framework needs to be built out. So here's for all of you in the room, online, and also in San Diego. We need your help. For many of you patients, I think or patients and families, Julia tells us all the time of that hard balance when you feel like you are trying to do your best to create a treatment, and anything that you ask for may be considered as onerous or may affect your ability to actually get that treatment to your loved one. I think that completely is understandable. At the same time, this is your data. This is the data from your family member or your child or yourself. This is a vital piece of building the framework for everyone else in this ecosystem and not just yourself, and you can advocate for that. The treatments may be very individual, but the learning truly is collective. So we have an N of 1 collaborative seminar series where we actively have both investigators as well as patients and drug development patients and who share their stories, as well as case conferences. All of this is shared in an open manner on our now bi-weekly seminar series that you're very welcome to. And by definition, rare diseases are not contained in any given country. We're global. They are global, and so are we. So we realize that we have to build this out in a global way from day one, and that's what we intend to do. So let me finish with one analogy, and this is adapted from an N of 1 evangelist, I would say, at Berkeley. His name is Fyodor Ernov. So here's El Capitan, and this is in Yosemite. It's 3,300 feet tall. The first time that this was climbed, it took 48 days over 18 months by 3 men. You may have heard of Free Solo and Alex Honnold. In 2017, he, as an insane person, or I would say insanely talented person, climbed it without any gear in 3 hours and 56 minutes. The process here is what's really important. Alex Honnold didn't just say, look up one day and just sent it, right? He divided up that 3,300 feet into 33 different pitches. Some of them are easy and some of them are hard. For the hard ones, he deliberately practiced while being roped in. And sometimes he did it dozens of times just to figure out how to do it in the most effective, safe, and rigorous manner. And I pose to you that this is actually the N of 1 problem in granite format, right? There's so many different pieces of what we have to solve along the way to climb that mountain. Some of them are hard. Some of them are kind of, or at least relatively easy. But what we need to do as a field is just practice every single one of them so that we can make better drugs and do it in a more efficacious way. So I'll leave it here and say that really no part of this problem right now is impossible. We need to work together. Things will be hard, and we need to make sure that we just collaboratively put our efforts into the right place. Fundamentally, like we say in the letter, our patients are waiting. We owe it to the rare disease patients to make sure that we do this in a responsible way. So with that, I'll acknowledge all of the amazing work being done at the N of 1 Collaborative, as well as the Chan Zuckerberg Initiative for their initial support. And there's a little QR code just to scan for the open letter. Okay, thank you.